Good morning, students. Myself Manoj Chotia from Raghunath Vidyalaya, Ratangad. Today we start our new topic, dormancy. What is dormancy? After completion development of seed in the plants, their seed growth stops. Their seed growth stops in some plants, even if the favorable environment conditions are available, but seeds are not germinated. While in other plants, seeds are germinate properly, but when the seeds are not germinate by its physiological structure or inactive stage, this phase of seed is called dormant stage, and this phenomena is called dormancy. Whenever a seed gets a favorable condition but it fails in germination, then it is called dormant stage, and this phenomena or process is called dormancy. Due to dormancy, seed germination is temporarily suspended for some time. Due to this, the seed remains viable for a long time. Suspension period range from 5 to 10 years in xerophytes such as acacia. Acacia has a 5 to 10 years time period of suspension. Lupine has 1000 year crop plant, as you show every year in our homes. 2 to 5 years the capacity to germinate. After 5 years they can't be germinate. But in this range 2 to 5 years they can makes their viability. And rhizophora and pea has the absent dormancy because they have viviparous germination, means their seed are germinate in their fruits. Now, types of dormancy, there is two types of dormancy: primary dormancy and secondary dormancy. When the mature seed do not immediately germinate, if their internal or external structure or functional condition are favorable, it is called primary dormancy. But whenever seeds are mature and due to change, whenever they will store it, so they can change their physiological activity and lose the capacity of germination, that is called secondary dormancy. Now we can see the factors affecting dormancy. These are the factors affecting dormancy. First, hard seed coat. Second, immaturity of embryo. Third, requirement of after ripening period. Fourth, require specific temperature and light. And fifth, presence of germinating inhibitors. First of all, hard seed coat. The germination is start by the activity of imbibition. But whenever the hard seed coat is found around the seed, then seed cannot absorb the water, so they can't be germinate, or we can say this is the mechanical resistance found in plants seed, just like amaranthus. Second, immaturity of embryo. In many plants the seed are dispersed before embryo mature. Hence, germination in these seeds is not possible unless the maturation of seed is not possible. Example, ginkgo biloba, nitum. Third, requirement of after ripening period, means a cold treatment is required before the germination of seed. These seed cannot germinate until they are not treated with a specific cold heat treatment. They are treated naturally in winter season, then after they can germinate. But whenever these treatment is not obtained by the seed, then they can't be germinate. So this is called requirement of after ripening period. Next, require specific temperature of or light. Many substances present in the pulp of pulpy fruits are able to inhibit germination of seeds, which are called some substances require specific temperature and light. As we know, all the plants need some temperature to germinate and some type of light, because in the presence of light plant get photosynthesis. So on these, if these factors or is not provided to the seeds, then they can't be germinate. Last, presence of germinating inhibitors. Many substances are present in the pulp of pulp fruit are able to inhibit the germination seed, which are called germinating inhibitors. Remember, these are the example of germinating inhibitor. Here 1 mark question is performed. Write the factors which are called germinating inhibitor. First, ABA, abscisic acid, coumarin, para-ascorbic acid and phenolic acid are the germinating inhibitors. Now our next topic is method of breaking seed dormancy. Methods of breaking seed dormancy. Different types of method are adapted to break the dormancy, which depends on the plant and dormancy factor. These are following. First, scarification. Second, chilling treatment. Third, exposure to alternate temperature. Fourth, light. Fifth, pressure. And sixth, use of growth regulator. First, scarification. In this method, the seed cell is weakened by breaking or scarification of seed coat. Many times these seeds are softened by putting into the solvent of dilute sulfuric acid and hot water or fat. So by this method we can weaken the seed coat after seed can be germinated. Chilling treatment. Vernalization is the one method by which the dormancy is also broken, or we can say whenever we treated seeds to cold temperature, then the dormancy is breaks. Exposure to alternate temperature. Whenever any seed not germinate, we treated them different types of temperature, means higher temperature and lower temperature, then the dormancy is breakdown. Like some seeds are germinate in the particular length period of time, that is called photoperiodism. So if we provide photoperiodism to seeds, then they can germinate. Next, pressure. The germinating capacity of some dormant seeds can be increased by keeping them into 2000 atmospheric pressure in between 18 to 20 degrees centigrade of temperature. And last, use of growth regulators. Those seeds whose dormancy near cold treatment, post mature period, presence of resistance of specific light, their dormancy can be broken by the use of growth regulators. Examples, remember them: thiourea, chlorohydrin, gibberellin and ethylene are the growth regulators which can break the dormancy. Our next topic is abscission. What is abscission? Means detached from the main plant of any part of plant, means leaf, fruit, flower, is called abscission. First of all, ABA, abscisic acid hormone makes a abscission layer between any two structure, means this is leaf and this is stem. Then ABA hormone make a layer which is called abscission layer and formation of this layer happens by the degeneration of middle lamella and cell wall by different types of enzyme like pectinase, cellulase, these enzymes dissolve the cell wall and middle lamella of the cell, and after ABA hormone make a layer which is called abscission layer. And after, whenever these plants get a high power of wind, then these structures are detached from the main plant. So this is called abscission, or falling leaves, flower and fruit separated from mother plant is called abscission. Abscission is a biological process that results from change in the cells of the basal part of the of these plant organ. At these places, mid pillars and outer walls of cell of a certain area become digestive by pectinase and cellulase enzymes, so that the cells begin to separate from each other due to the breakdown of the middle lamella and ribs. This tissue of this area become soft and weak, and abscission layer is formed at this place. The cells below the abscission layer constitute partially degenerate of the cells that form a protective site or a detachment area. As a result of heavy wind below or rain, a plant part break down from the place of abscission layer and separate from the plants. Abscission due to the change in the hormone balance. In this process, abscisic acid play an important role. So this is abscission. Now in this chapter only two topics are remains: vernalization and dormancy, which we will study tomorrow.